Hi, my name is Stephen David, and today I'll be presenting some work from my lab at Oregon Health and Science University that we've performed in collaboration with the group of Nima Mesgarani at Columbia. And the question we're looking at today is, can deep learning produce a general model of natural sound encoding by auditory cortex? So what do I mean by encoding model? The goal we have here is to understand how the brain represents and computes information about the sound that it encounters in the real world. So if I use an electrode to record activity of neurons in auditory cortex during the presentation of sound, the timing and magnitude of neural action potentials that we record will reflect some information about the sound that's being presented. And when you use an encoding model, what you're trying to do is replicate those computations. So if you provide the stimulus as an input to the model, and it accurately can describe the function of the system, then it should accurately be able to predict the output of the neurons in response to those same arbitrary sounds. And so a good model is one that sees a strong correspondence between the predicted and actual response. So there have been many advances in the performance of encoding models, but there are still important gaps in our knowledge, and I'll be talking about some new approaches that we've taken to this problem today. And so developing more accurate encoding models can be valuable for a number of reasons in neuroscience. At a very basic level, understanding central auditory processing gives us a better understanding of the brain systems that can be affected by hearing loss. And the healthy auditory system can also operate in very challenging and complex acoustic environments. And so if we have a better understanding of how the healthy brain works, we can use that to develop algorithms to have sort of better performing hearing aids and other auditory prosthetics. So given that sort of big picture motivation, I'll be talking about three things today. First, I'll motivate the use of this new method, deep neural networks, to study auditory coding. Then I'll show that these DNNs, in fact, perform better than standard widely used encoding models for describing activity in auditory cortex, and particularly the time-varying activity that occurs during presentation of natural sounds. Then finally, I'll get at a challenging question that we encounter with DNNs, which is how to interpret them. And so I'll spend some time showing that some methods we've developed can be used to understand what it is that these new models are doing. So as I mentioned, there's been substantial effort previously to build encoding models in the auditory system as well as other sensory systems. And in the auditory system, the majority of models are based on the idea of spectro-temporal representations. So it's well established that in the cochlea, different populations of receptors respond to sounds at different frequencies. So you can model this peripheral activity as what's called the sound spectrogram, which is shown at the bottom here, where each row shows the sort of level of activation in response to a sound at a different frequency. And as you can see with this example segment of speech, natural sounds contain complex spectral patterns. And the job of the auditory system is to recognize and discriminate these patterns that have different behavioral meanings. And so what you might call the standard model for sound coding in auditory neurons is the linear-nonlinear spectro-temporal receptive field, often called the STRF. According to this model, each neuron is sensitive to stimuli in a particular range of frequencies and time lags. This is illustrated here by the heat map in the upper right, where red areas indicate features that increase spiking activity, and blue areas, when they're present, indicate regions or features that suppress spiking activity. Using the STRF, the activity of a neuron can be predicted by convolving this linear filter with the sound spectrogram. The output of the filter is followed by what's called a static nonlinearity, which accounts for the fact that neurons cannot produce negative action potentials. And so this is used in the auditory system, but it's also used more widely. This general formulation is used more widely across sensory systems, and it's often referred to as the linear-nonlinear or LN model. And in the case of this neuron, you can see that the LN model is doing a pretty good job. So the panel at the bottom shows the prediction of the LN model in red, and you can see that it corresponds pretty well to the actual response that was evoked in the neuron that was being studied here, which is shown in blue. So the LN-STRF is appealing because it's relatively easy to understand what's going on. You can look at the linear filter and you can see what sound features are encoded by the neuron. And so this can be used, for instance, to measure the best frequency or the bandwidth of the responses of the neurons. And using this approach, a number of studies have developed basic models of what's going on in the auditory periphery and how sounds are represented at that stage of processing. However, the problem with the LN models becomes a little bit bigger in more central areas, especially in auditory cortex, because what happens is these models don't predict everything that the sounds actually produce. And so you can see that in this example where, again, we have an STRF. The color map has changed here, but the red and blue areas still indicate excitatory and inhibitory tuning. And in this example, we use the LN model to predict the response to, from a different neuron to predict response to a natural vocalization. And in this data set, the actual response is plotted in gray. And you can see that there are periods of brief activation associated with the onset and offset of different sound features. And the LN model prediction is overlaid in orange. You can see in this case, it predicts the coarse activation in response to different sound features, but it fails to predict this fine temporal structure. And across auditory cortex, the performance, the accuracy of prediction by the LN models is limited. So we can quantify prediction accuracy with the correlation coefficient, or R value, between the predicted and actual time-varying response. Using this metric, a maximum value of 1 indicates a perfect prediction, and the R squared value indicates the fraction of variance explained in the neural response. And so in one study, after accounting for a number of experimental factors that can limit model performance, we found that the sort of maximal performance on average across neurons in auditory cortex explains about 40% of response variance. And so there is room for improvement over these models. And so this problem is well known, and a number of efforts have been made in past decades to develop models that can perform better than the STRF. And so I don't have time to go into all the details of these models because there are quite a few, and they've taken different approaches to extending the STRF, either using ideas of different biophysical properties present in the system, or using more general approaches to expand beyond the linear representations to various nonlinear interactions that can be observed between features. And so one class of model that has grown very popular recently in a number of different applications is a new machine learning approach called deep learning, or using deep neural networks to model these responses. And so these models have been developed and applied largely in the visual system so far, and with a little bit of work done in the auditory system as well. And the question here today is, can we use these approaches to describe the activity of single units in auditory cortex? And so just to kind of point out again and emphasize the kind of differences between these different approaches to modeling, you can think of the LN model as kind of being at one end, or near one end of the encoding model spectrum, where they're relatively simple. And so while they don't generalize to a large number of stimuli very effectively, they are relatively easy to interpret. And these more complex models that have been developed are generally able to predict activity in a wider range of circumstances, but they're also more complex to fit, and they require more data for fitting. And these deep networks, or sort of the general class of artificial neural networks, lies at kind of the extreme case, where they have the most generalizability, but also are the most complex. And with this complexity becomes a challenge of interpretation. And this is something that is particularly important for understanding really the computations that are taking place, because these models are very large and complex. It's difficult to tell exactly what information they're representing in the neural computations. So, as I mentioned, the DNNs have been applied most successfully and extensively in the visual system. And this diagram here, taken from the work of Yemens and DiCarlo, illustrates why it's very appealing as a model for sensory processing, particularly in sensory systems where there is an idea that information is processed by a hierarchical sequence of sensory areas. And so each dot here in these squares at the top indicate a cluster of simulated neurons. Each one really is effectively an implementation of an LN model. And so you have these layers of LN neurons, each with slightly different tuning, that feed into subsequent layers. And what has been demonstrated just empirically is that if you have these large networks of linear-nonlinear units, that you can account for very complex input-output relationships. And what appears to also be useful is that the laminar structure, the processing that happens at each stage, seems to roughly parallel the transformations in representation that occur in the visual system. And so what do I mean by that? Well, when you record data from a very central visual area, in this case, inferior temporal cortex, which is associated with high-level object recognition, what you find is that you can train a DNN on a computer vision task, so to discriminate different categories of images. And if you take the output of that deep network, you're able to use a linear weighted combination of those outputs to predict the activity of a large number of neurons in inferior temporal cortex. If you simulate the activity of earlier layers in the network, which have responses that look like earlier processing stages in the visual system, they aren't able to predict activity as well, as you can see in the performance comparison between the early stage models at left and the late stage models at right. So this shows that especially in central areas, that deep networks may actually be very powerful and very useful for explaining neural activity. But as I mentioned, these are very complex models with many parameters, and interpreting them has been challenging. So far, what's been most successful is this approach, which simulates the activity of the deep network to try to determine what you might call optimal stimuli for the neuron. So that you can simulate a very strong response from the network, and you can backpropagate the effect or backproject the activity to see what pattern or patterns produce that activity in the output. And this shows some examples, again, getting at this idea of laminar hierarchy, where you can backproject activity from either earlier layers, which are shown at left, which produce features that are related to features that have been proposed to occur at mid-level representations in the visual system. And if you backproject the late stages, what you get are features that actually look like visual objects that are believed to be associated with activity in these central processing areas. DNN analysis has not been applied as much in the auditory system, but recently our collaborators used the same general approach to study encoding in human auditory cortex using LFP data recorded from human epilepsy patients undergoing monitoring for treatment. And they used a similar architecture of these layers of LN units as are illustrated in the bottom of this picture here. And what they found is that when they compared performance to a traditional LN model, as is shown in the scatter plot at the left, that the DNN performed consistently better both in primary auditory cortex, which is Heschel's gyrus or HG, as well as in superior temporal gyrus, which is a tertiary or higher order representation in human auditory cortex. And so the question we had today was, can we use the same modeling approach to understand the activity of single units in the auditory cortex of animals in a similar experimental condition? So the next question here is, well, can we describe the time-varying activity of neurons in auditory cortex? Briefly, to describe our experimental approach, we were presenting a large library of natural sounds, about 600 different 1-second samples, as well as a set of repeated sounds used for model validation. We presented these to awake, passively listening ferrets using free field stimulation contralateral to the recording site. And to collect single unit activity, we used laminar probes, 6428 channel probes from UCLA that permitted simultaneous recording of somewhere between 20 and 50 single units in an experiment. We could identify lamina in different cortical fields, although we're not talking about the specific laminar patterns today. So in the ferret, there's a hierarchy of auditory areas that's roughly matched to those that of primates. Auditory stimuli come into the midbrain. They're processed at several stages, including the inferior colliculus, then pass through primary auditory cortex, or A1, and then some non-primary areas called the belt. And in the ferrets, it's called area PEG. Eventually, that information gets, which are believed to guide behavioral decisions. The data I'm talking about today was recorded from two areas, A1, which is primary auditory cortex, as well as this belt region termed PEG. So we presented the same stimuli in each experiment, the same large library of natural sounds. And so, as I mentioned, we could record from multiple neurons in one experiment. But then what we could also do is construct a pseudo population by combining the data from all of these experiments into one large data set that merged all of these different recording sites. And again, we had multiple sites from both A1 and the secondary area, PG. And the details of the models that we tested, we used a standard linear-nonlinear model as a baseline, and then we explored a number of different DNN architectures to kind of build on the previous results. We used a small DNN to analyze the activity of individual single units that were recorded in the study. And this diagram here just shows roughly the layout of the layers and arrangement of the linear and nonlinear units. The linear units were linear filters at the input or dense units at later layers. And the nonlinear in this case is just a rectified a linear rectifier, which we chose for reasons that may become apparent when we talk about visualization in a few slides. So in addition to this, what we call a single unit DNN, we also developed what we call a population model, where we had a similar architecture, but instead of just predicting the activity of a single unit, we had a large output layer that combined the previous layer with different weights in order to predict the activity of each of the neurons in the population. And what we found is that this could leverage the activity of a large number of neurons into a common representation that kind of took advantage of this additional statistical power to get a better prediction accuracy. And we used two different architectures for the input layers. What we call the 1D convolution was mimicking the classic STRF model, the filters that I discussed already at the input. And then we also used a 2D convolution, which is more analogous to the models that have been used in the visual system and auditory system previously. So we evaluated a large number of models that varied in the size and number of layers. I don't have time to go into all the details today, but actually, as we found, the performance of these models was very similar, independent of the architecture. So those details may actually not be so important. For fitting, we used the neural encoding model system library, which was developed as part of the BRAIN initiative that supports this project. And the models were fit using backpropagation implemented in a TensorFlow backend. So how did it do? Well, here's an example of the neuron that I showed you previously, where the actual response shows the sort of complex temporal dynamics. When we predict responses with this LN model, it fails to predict those responses. So then when we used a DNN model, again, implemented here, this was using a 1D convolution in the architectures that I mentioned. It was able to capture quite a bit of the detail in the temporal response, as you can see in this purple plot that's overlaid at the bottom. And across the population of neurons in primary auditory cortex, we found that there was a consistent improvement for the DNN over the LN model. So again, this is one architecture that performed particularly well, and the black dots indicate neurons for which the prediction accuracy was significantly greater, according to a t-test, for the DNN over the LN model. One thing that I mentioned was striking about this was that the performance of the different DNN architectures was actually quite similar. So here are two more example neurons, again plotted with the stimulus spectrogram at the top and the simultaneous activity of the recorded in the two panels at the bottom. Gray shows the actual PSTH, and the performance of the LN model is shown in orange. The top panel, you can see that, again, the LN model isn't able to capture the fine temporal structure of the response, but does get the overall activation by the different stimuli. The bottom panel shows a different case where the LN model was just unable to capture, it captured some fine temporal dynamics, but couldn't capture the relative structure of the gray peaks. For instance, that initial onset response, it underpredicted quite substantially. So when we used, again, this is the 1D convolution model that I mentioned, you can see that it does a much better job capturing the responses in both cases, capturing both the fine temporal structure in the top and those large onsets that we see at the bottom. What was very striking, though, is when we tried different architectures. For instance, here's a 2D convolution. We saw nearly identical results. So a much different kind of configuration of artificial neurons, but we got a very similar prediction in the end. And this is what we saw across the population as well. So at the left, I'm showing, again, the performance of the DNN relative to the LN model. Then the middle panel just shows the performance of these two different DNN architectures. And there are a few neurons where one model performed better than the other, but overall they're actually predicting with very, very similar accuracy. And we can also compare what we call the equivalence of these different models by simply computing the correlation coefficient between the predicted responses of the different models. And so if we do that for the LN model versus one of the DNN models, that's plotted in the orange histogram, you can see that the correlations are generally relatively low, lower than 1. But in the case of the two different DNN architectures, which are plotted in blue, you can see that they're significantly higher and often very close to 1, which indicates that the predictions are actually identical. So, as I mentioned, we compared performance of a large number of different formulations of the DNN architecture. And this Pareto plot shows the average performance, the median performance across all the neurons that we reported from in A1. And the x-axis shows what we call the complexity of the model, which is basically the number of free parameters that were required per neuron and the model fits. And what you might note most prominently is that the model at the bottom is the performance of the LN model, which peaked at a core 5. And that didn't matter if we analyzed each neuron separately or analyzed them as a population model, similar to the population models that we were using for the DNN. On the other hand, all the DNN models performed quite similarly, and this includes, again, both the 1D convolution as well as the 2D convolution architecture, and a number of different variants that are sort of schematized here. One thing to note is that the single neuron DNN achieved almost as good performance, but required generally a larger number of parameters per neuron in order to achieve the same level of performance. We performed the same analysis on the data that we collected from non-primary cortex, or PEG, and observed basically the same pattern, where the DNNs performed similarly and substantially better than the LN model. Although in this case, while everything was a little bit worse than in A1, the relative improvement was actually greater for the DNNs. So where we saw about a 30% improvement in A1, we saw close to a 40 or 45% improvement in PEG. So another interesting observation that we made with the DNN models was that they were able to generalize quite effectively to new data. So as I mentioned, we combined data from a number of recording sites into a pooled population in order to fit our models. But in a subsequent study, we refit the models excluding one site each time. And so we could train the entire model. Then using this pre-trained model, we could fit only the output layer using data from this held out site. And when we did this, this is data from A1, what we found is that the different DNN models performed nearly identically. You can see the strip plots compare performance of the held out and the matched models. And you can see that the mean performance was nearly identical for all the DNN architectures. But you'll note with the single unit DNN model, where we performed an analogous transfer where we fit the model to one cell and then fit the outputs to a new cell, we saw that the held out models did much worse than the original model, showing that by fitting these populations together, we're able to analyze this quite effectively. So finally, I wanted to briefly describe the approaches that we're using to understand what computations are being captured by these models. So we can look at these models in their entirety. So this is an illustration of one DNN fit using the 1D convolution approach. In the top, we see all the filters, which look like LN models. And you can see that they're connected by these lines through a sequence of layers. And the color or the brightness of the line indicates the strength of that weight. So you can see that there's a lot going on, and we can actually look at the prediction of the model on the right. We're comparing the population prediction to the actual response that we saw in the bottom panel. And you can see that there's very roughly a correspondence of these, the predicted and actual responses. But really understanding what's going on here is tricky because there's so many free parameters and so much going on. So what we did is use an approach that was developed by our collaborators and applied previously to the human LFP data. And that took advantage of a relatively simple but important observation of these networks, which is that at any given moment in time when you're presenting stimuli, a number of the static nonlinearities, these rectified linearities that I mentioned earlier, are below threshold. So basically, the input is not driving their outputs, and so they're effectively absent from the activity of the network in this particular state. And so what we can do is we can essentially remove those nodes, and what's left is the activated nodes, which are all performing just simple linear transformations. And so the net activity of the DNN at any given moment in time can actually be collapsed into a single linear filter, which looks just like the STRF that we presented previously. But it's important to remember, of course, that this is a dynamic STRF. So the specific STRF will change depending on the particular stimulus that you're presenting. And so just to get a very simple intuition for this, you can think of the stimulus-response relationship for a neural network or any nonlinear system as this, in this cartoon example, some curve that's not flat, right? That's a nonlinear function. And if we look at some small regime of stimulation, for instance, the yellow or the blue areas here, the performance within that regime can be approximated using a line. And so what we're doing when we compute a DSTRF is effectively fitting this locally linear model to the activity in this sort of restricted sensory context. And so what does that look like with the data that we have? So here's that first example that I showed you, and there's quite a bit going on here. But what you might recognize at the top is the stimulus, and then the prediction of the LN model, and then the DNN model in purple. And what I'm showing in the bottom row are dynamic STRFs that were fit, again, using at each point in time that's indicated by those dashed red lines. And so what you might notice is that the pattern of activation, the red and blue areas that indicate excitatory and inhibitory tuning, are changing from moment to moment. And so what this illustrates is that while the LN STRF shows a single pattern of activation, that actually this neuron is responding to different spectro-temporal patterns at different points in time. But if you look carefully at this data, what you'll see is that it's not completely random. So the DSTRFs are changing, but they're actually changing in somewhat repeatable ways. And so you can see that by performing a principal component analysis, or PCA, on the DSTRFs. And the first three PCs are shown at the bottom here. And what they show is that there is a relatively small set of features that explain the variations that we're seeing in the DSTRF across time. So we use this DSTRF analysis and then perform PCA on the cells in our data set. And here are just 10 examples showing at the top the first PC, the largest PC, and then the bottom two rows show the second and third PCs for those same neurons. And again, there's a lot to look at here, and this is an area that we're actively exploring to try to understand what information is captured by these neurons. But just very roughly, one thing you might notice is that the relative contribution of these PCs can vary from neuron to neuron. So the brightness of the DSTRF indicates the relative variance explained in the total DSTRF by each principal component. And if we compute that value per neuron, what we see is that, as expected with PCA, the first component is always the largest, but the relative magnitude of that component varies substantially from cell to cell. And so one thing that you can predict is that that these more complex spaces that are captured, that require a larger number of principal components to capture, are going to correspond to neurons that showed a greater benefit of this more complex DNN model. And in fact, that's the case. So at the right here, we're plotting the fraction of variance explained by the first principal component in the DSTRF against the relative improvement, so the difference between prediction performance for the DNN and the LN model. And what you find is that for the cells that show the greatest performance, increase in performance at right, the relative fraction explained by the first PC is smaller. And so indeed, these more complex dynamic STRFs correspond to cells where the DNN model is showing the greatest improvement. And so we're now using this approach to analyze data from secondary cortex as well, but unfortunately I don't have time to get into that today. So just briefly to recap what I talked about today, first, what we have shown is that deep neural networks, or DNNs, can account for time-varying activity of single neurons in auditory cortex consistently and often substantially better than standard linear-nonlinear models. What was Also quite striking is that the performance of the DNNs did not depend on the details of the architecture. And so we could use a large number of different formulations and get very similar results. However, with this population model, one exciting thing that we observed was that we were able to use these prefit models to new data sets. And that opens up a number of possibilities where we may be able to use them to analyze data from experiments where the amount of data is often limited. For instance, in behavioral studies where the number of trials that you can get in a particular behavioral condition is often limited, and it's often been difficult to use encoding model analysis for those types of studies. And finally, we're using this method of dynamic STRF measurement to describe the space or the range of spectro-temporal patterns that describe tuning. And we can use this as a measure of complexity to compare representations in different brain areas. So with that, I wanted to make sure to thank everyone who contributed to this project. Jacob Pennington is a student in my lab who really did the bulk of the analysis and modeling. Also, of course, Nima Mezgarani and Manua Kashishian at Columbia were instrumental in developing these methods, in particular the DSTRF analysis. I also wanted to mention a couple students in my lab, Mateo Lopez Espejo and Charlie Heller, who were instrumental in collecting the data that we analyzed here. Also, the funding from the Brain Initiative, in particular for this project. I did want to mention that all the software that we use for this analysis is available as part of the Neural Encoding Model System, or NEMS. That's available on GitHub. So please feel free to contact me if you have any questions about that. And finally, thank you for listening.